Welcome to the Deepdive Lab, where we dive deep into fascinating science stories filled with surprising discoveries, unexpected twists, and, thought-provoking ideas.
Yeah, it's great to be here.
So, to start us off, I want you to think about something a little strange. Right now, as you listen to this, you are carrying around anywhere from 8 to 12 chronic viruses inside your body.
Which I know sounds a bit alarming when you hear it like that.
Right, but you don't feel them, and you probably don't even know they're there. I mean, for
Yeah.
the vast majority of your life, these viruses just sit in a state of deep latency.
Exactly. They basically weave their genetic material into your long-lived cells, quiet their own gene expression, and, well, they essentially go to sleep.
And they just stay there, like
Yeah.
maintaining this very delicate, I guess, millions of years old truce with your immune system.
Yeah, a very careful truce. So viruses like Epstein-Barr, which causes mono or cytomegalovirus, usually known as CMV, they establish these lifelong residencies in you.
And the established medical dogma, like the textbook rule for these latent infections, has always dictated a very specific rule of engagement, right?
Right. The old belief was that they only wake up when the host's immune system is severely suppressed.
Like when someone is going through a really intense medical treatment.
Exactly. We typically see this kind of reactivation in patients undergoing aggressive chemotherapy or those on immunosuppressive drugs following an organ transplant. The guards are down, so the virus wakes up.
Which sets up the central paradox of our deep dive today. We're looking at a landmark August 2026 paper published in the journal Nature based on the IMPACC study.
And this study tracked severe COVID-19 patients.
Right, but here's the thing. Severe COVID-19 does not suppress the immune system. It triggers the exact opposite.
It absolutely does. You get massive immune overdrive. It's a profound state of hyperinflammation.
So doctors in intensive care units were catching glimpses of these old, dormant viruses springing back to life in COVID patients. And that completely defied the textbook understanding of virology.
Completely. I mean, if the immune system is firing on all cylinders, swarming with inflammatory cytokines and highly active T cells, how on earth does a sleeping virus manage to slip past the heavily armed guards?
Yeah, I mean, it feels like waking up to rob a bank while the lobby is literally filled with SWAT teams. It makes no sense.
It really doesn't. And that glaring contradiction is what drove the IMPACC investigators to launch this massive, highly coordinated study.
Because they needed to know if these awakened viruses were just, innocent bystanders getting jostled awake by the physiological chaos of COVID-19.
Or, and this is the scary thought, are they actively participating? Like pulling the strings and driving the severity of the disease themselves?
So to figure that out, they went big. They tracked 11,154 hospitalized COVID-19 patients across 20 different U.S. hospitals.
And they didn't just check their blood work once on admission. They monitored them continuously for up to a year.
A full year. That's a massive amount of data.
The scale of the clinical tracking is impressive, but the analytical approach is what actually makes this a landmark paper. They deployed something called multi-omics.
Multi-omics. Okay, break that down for us.
So rather than relying on just a single biological metric, they layered multiple distinct data sets on top of each other to get a complete picture.
I mean, it sounds kind of like the difference between looking at a static photograph of a factory versus simultaneously having the architectural blueprints and the live security footage of the machinery operating and the daily accounting ledgers all laid out on the table at once.
Oh, that captures the methodology perfectly.
That's awesome.
So the blueprints in this case are the RNA transcripts, which reveal exactly which genes, both human and viral, are actively being turned on.
Right. And the machinery.
The machinery is the proteomics. That details the physical proteins that are actually executing those genetic orders.
And the ledgers.
The ledgers are the metabolites. That's the chemical exhaust left behind by all this cellular activity.
Wow. So they track this multi-omic data across the blood, inside nasal swabs, and even deep within the lungs using endotracheal fluids.
They inspected the entire biological supply chain across multiple organ systems. And what they found completely upends that innocent bystander theory we talked about.
Because the timeline of when these viruses woke up wasn't just a random, chaotic free-for-all.
No, it was highly choreographed. The temporal dynamics were incredibly distinct.
So who wakes up first?
Epstein-Barr virus, or EBV, along with a massive family of viruses called Anelloviridae. They reactivated extremely early in the disease progression.
Like, how early?
Their activity peaks between days 1 and 8 of hospitalization.
Wow, that is fast.
It is. In fact, when analyzing the admission data, researchers noted such high titers of EBV antibodies that it strongly suggests the virus begins reactivating before the patient even steps foot in the hospital.
Wait, really? It's already awake before they even get admitted?
Exactly. But then you look at the schedule for other latent infections, like CMV or HSV-1, which is the virus responsible for cold sores.
Oh, right.
They don't peak until much later, around day 22, and they primarily reactivate within the respiratory tract rather than systemically in the blood.
So, if reactivation were merely a passive response to the general stress of a severe infection, you'd expect just a uniform pan-viral awakening. Everybody wakes up at once.
Right. But this staggered shift change, EBV clocking in on day 1 and CMV waiting 3 weeks to emerge, that points to a targeted systemic alteration of the body's viral ecosystem.
And scientists have a specific name for this state now, right?
Yes, they're increasingly referring to it as dysvirosis.
Dysvirosis, which, we hear a lot about dysbiosis in the gut microbiome, like when the balance of good and bad bacteria gets thrown off and causes systemic health issues.
Exactly the same concept.
So dysvirosis is just applying that same ecological framework to the virome. It's a complex dysregulation of the viral flora we carry around.
And investigating that dysvirosis led to one of the most significant paradigm shifts in the paper, centered squarely on that viral family you mentioned earlier, Anelloviridae.
Yes, Anelloviridae. This is a virus family found in, 80 to 90% of the general population.
Yeah, it is nearly ubiquitous. Almost everyone has it.
And for decades, the scientific consensus largely dismissed it as a harmless passenger virus. Like, it was assumed to just float along in our bodies without causing any structural or immunological damage at all.
Right. But the multiomics data showed it reactivating aggressively in these COVID patients.
And the clinical records of those specific patients are where the old medical dogma officially shatters, because only 17.4% of the patients experiencing this Anelloviridae reactivation were on any form of immunosuppressive medication.
Let that sink in. Over 80% of them had fully active, highly inflammatory immune responses.
So what is the actual biological mechanism here? Like, if a suppressed immune system isn't what wakes them up, how does severe physiological stress act as an alarm clock?
Well, it comes down to how viruses hide in the first place. Latency is maintained by complex epigenetic silencing.
Meaning the body kind of locks it away.
Yes. The host cell basically wraps the viral DNA up tightly so it can't be read. But inflammatory cytokines, the very same signaling proteins your body pumps out to fight COVID-19, can inadvertently act as chemical peaks.
Oh, wow. So when a cytokine storm hits...
When a cytokine storm hits, certain transcription factors meant to activate human defense genes end up binding directly to the viral promoter regions. The inflammation itself unspools the viral DNA and triggers the reactivation.
That is wild. The immune system's own alarm bells are accidentally wiring the explosives for the dormant viruses.
It is wild. And it forces a complete reevaluation of how we handle severe respiratory infections. The researchers immediately recognized they needed to map the specific biological damage these awakened viruses were causing.
To see if they were actively amplifying the host's hyperinflammation.
Exactly.
So let's track that damage profile, starting with the early riser, Epstein-Barr. The data links EBV reactivation to severe spikes in inflammatory cytokines, specifically interleukin-6 and interleukin-10.
And clinically, this correlated with a much higher likelihood of the patient going into shock and requiring intensive care.
But wait, looking closely at that cytokine profile, something doesn't add up. IL-10 is widely known as an anti-inflammatory cytokine, right?
It is.
It's basically an off switch meant to calm the immune system down. So if EBV is triggering IL-10, why are patients experiencing such extreme inflammatory shock? Shouldn't the immune response be quieting down?
That is where EBV reveals its true evolutionary brilliance.
Uh-oh.
The IL-10 flooding the system in these patients isn't entirely human. EBV actually carries a gene that allows it to manufacture its own viral version of interleukin-10, known as VIL-10.
Wait, it's synthesizing a counterfeit off switch?
Yes, a highly effective counterfeit off switch. This viral IL-10 binds to the same receptors on human immune cells that natural IL-10 does, but it functions differently. It specifically paralyzes the immune cells that would otherwise hunt down the EBV, essentially blinding the localized defense mechanisms. But because it's a counterfeit, it totally fails to trigger the broad, systemic calming effect that natural IL-10 provides.
So the localized guards are paralyzed by the counterfeit signal, allowing EBV to just replicate unchecked.
Right.
While the broader systemic cytokine storm caused by COVID-19 continues to rage totally out of control, it is actively sabotaging the immune system's communication network.
It's incredibly devious.
Mhm.
And while EBV is manipulating the communication lines early in the infection, CMV takes a completely different destructive path weeks later.
Right, around day 22.
Yeah, when CMV reactivates, the multiomic data shows it driving massive metabolic disruptions, specifically targeting the body's filtration system.
The kidneys.
Yes. The researchers identified severe renal impairment. They tracked elevated urea and specifically noted a massive spike in a metabolite called TMAP. Okay, what is that? TMB is a highly specific metabolic waste product. In a healthy individual, the kidneys constantly filter TMP out of the blood and excrete it.
So if it's building up.
When you see elevated levels of TMP accumulating in the blood plasma, it's a blaring chemical warning light that the kidney's structural filtration mechanisms are failing under stress.
Oh, man. And CMV reactivation was heavily correlated with this specific organ damage.
It was.
So we have EBV scrambling the immune signaling and CMV actively degrading kidney function. But the multi-omic data also revealed a shared mechanism of damage across the reactivated virome, right?
It did, particularly within the Herpesviridae family, which includes both EBV and CMV. They were causing a severe depletion of a molecule called 6-bromotryptophan.
6-bromotryptophan, which is incredibly dangerous to lose in the context of a severe viral infection.
Yes, because 6-bromotryptophan basically acts as a potent rust-proofing agent for your cells.
Defending against oxidative stress.
Exactly. When your immune system goes into overdrive to fight a primary infection like COVID-19, it requires massive amounts of metabolic energy.
Makes sense.
And a byproduct of generating all that energy is the release of reactive oxygen species. These are highly volatile molecules that will literally tear through cell membranes, mutate DNA, and denature proteins if they aren't neutralized.
And 6-bromotryptophan is one of the key molecules the body relies on to mop up that cellular rust and protect the tissue integrity.
Precisely.
So the massive metabolic demand of fighting the primary COVID infection is already producing record levels of oxidative stress. And right when the body needs its rust-proofing agents the most, these awakened latent viruses actively strip them away.
Yes. They leave the host cells entirely vulnerable to structural collapse.
Wow. The cascading failures just become inevitable at that point.
Yeah.
Treating the primary COVID-19 infection isn't enough if the latent virome has already dismantled the body's metabolic defense.
Then hijacked its cytokine signaling.
Exactly. Which brings us to, I think, the most critical consequence of this research. What happens to you after the acute phase?
After they survive the shock.
Right, after the kidneys stabilize and you're finally discharged from the hospital.
Well, the IMPACC study didn't stop at the hospital doors. They tracked these patients deep into their convalescence, analyzing them 3 to 12 months post-infection.
And they categorized the survivors using patient-reported outcomes, or PROs. Basically, they grouped them by how they were actually functioning in their daily lives.
Yeah. Some patients reported minimal deficits and essentially returned to baseline. Others struggled with specific cognitive deficits. But the researchers paid special attention to what they classified as the physical long COVID group.
Physical long COVID group.
These were individuals suffering from persistent physical disability, profound chronic fatigue, and a severe inability to exert themselves physically without triggering just a total exhaustion crash.
And the investigators wanted to know if the dysvirosis observed in the ICU was casting a shadow months later.
They did. And interestingly, acute EBV reactivation didn't cleanly map to this specific long COVID phenotype in this early pandemic cohort.
But the previously dismissed passenger virus did. Anelloviridae.
Yes. Patients trapped in that physical long COVID group demonstrated significantly higher rates of ongoing Anelloviridae reactivation up to a year after their initial infection.
So the virus had not gone back to sleep.
No, it hadn't. And it wasn't just silently replicating either. The data directly linked this persistent Anelloviridae activity to an ongoing biological process called neutrophil degranulation.
Neutrophil degranulation. Okay, neutrophils are frontline immune cells, right?
Yes. They are packed with dense granules full of highly toxic enzymes and proteases, which they use to destroy invading pathogens.
And degranulation.
Degranulation is the process of dropping those toxic payloads into the surrounding tissue. It is designed to be a short, intensely targeted burst of biological warfare.
So if Anelloviridae is constantly triggering neutrophil degranulation months after the primary COVID-19 infection is gone.
It means the immune system is relentlessly carpet bombing the body's own tissues.
That's brutal.
It is. The chronic release of those toxic enzymes heavily damages the endothelial cells lining the blood vessels, severely impairing the body's ability to deliver oxygen to muscles and organs.
And that biological mechanism directly explains the profound physical exhaustion and inability to exert energy that defines that specific long COVID phenotype.
Exactly. The harmless passenger virus is actually grabbing the steering wheel and driving chronic systemic inflammation.
Which introduces a completely new framework for how we practice critical care medicine. Because, the real tragedy here would be identifying this massive secondary wave of viral damage and having no way to fight it.
Right. But the researchers point out that the infrastructure to prevent this already exists. We don't need to spend a decade developing novel gene therapies or experimental tech to address this.
We don't.
No. Every major hospital system in the country is already equipped with standard qPCR testing capabilities designed to detect these exact latent viruses.
And we already have a deep arsenal of FDA-approved antiviral medications that specifically target viruses like the herpes viridae family.
We do. The bottleneck hasn't been a lack of pharmacological tools. It has been a fundamental blind spot in our medical perspective.
Because we assume these viruses only reactivated during severe immunosuppression.
Exactly. Doctors simply weren't looking for them during hyperinflammatory events like severe COVID-19.
So this paper essentially provides the blueprint for a major clinical shift. If you're admitted with a severe respiratory infection, they shouldn't just treat the primary pathogen.
They need to run a targeted panel to monitor the virome.
And the moment the multiomic data flags that EBV is throwing counterfeit off switches or CMV is beginning to stress your kidneys, they intervene early with existing antivirals.
You force those viruses back into latency before they can strip the body's rust-proofing, potentially heading off both the acute organ failure and the downstream cascade that leads to long COVID.
It demands that we stop viewing human infections in a vacuum. A novel virus does not enter a pristine, empty host.
No, it invades a highly complex, interconnected ecosystem.
And when a primary infection destabilizes that delicate virome, medical intervention really must treat the entire ecosystem, stabilizing the latent threats before they become active participants in the disease.
That's the key takeaway.
So distilling this massive multi-omic investigation down, there are 3 core insights you need to take away from this deep dive. First, your body harbors a hidden virome-dormant viruses that can be violently awakened not just by a suppressed immune system, but by the sheer physiological stress and inflammation of a severe infection.
The old medical rules regarding latency have fundamentally changed.
Right, and
Second, these viral reactivations are highly orchestrated events that cause specific, targeted damage. Whether it is EBV manufacturing counterfeit signaling proteins to blind localized immunity, or CMV severely compromising the kidney's metabolic filtration, these viruses actively amplify the severity of the primary illness.
And third, the viral ecosystem we leave behind when we recover from the acute illness might actually dictate our long-term health.
Yes, the chronic, low-level reactivation of previously ignored viruses like Anelloviridae appears to play a direct role in driving the relentless immune crossfire and physical exhaustion seen in long COVID.
It really forces us to confront how much of what we consider to be the normal aftermath of a severe illness is actually the quiet work of these latent viruses.
Science is just a constant process of unlearning, and realizing that harmless passengers might actually be driving the pathology requires a total reassessment of human biology.
It absolutely does. Which leaves us with one final, deeply provocative question. This study focused on what happens when a massive physiological shock to the system, like a severe COVID-19 infection in an ICU, orchestrates this coordinated uprising of dormant viruses.
Right, in an extreme scenario.
But if inflammatory signaling and physiological stress alone are the keys that unlock viral latency, what else is pulling their strings?
That is the million-dollar question.
Consider the cumulative impact of everyday life. Could a prolonged period of intense anxiety at work, or a string of sleepless nights, or even fighting off a mild winter cold, generate just enough inflammatory cytokines to briefly wake these sleeping giants?
It's entirely possible.
And if they're briefly waking up, throwing a few counterfeit signals, depleting a fraction of your cellular rust-proofing, and then quietly slipping back into latency, how is that invisible cycle subtly shaping your energy levels, your cognitive clarity, and the very way your body ages over the decades?
Understanding that daily, silent dialogue between our immune system and our virome is undoubtedly the next great frontier in immunology.
Thank you for taking this deep dive into the hidden world inside all of us. If you want to keep exploring the unseen forces that shape our biology and our world, be sure to follow and subscribe. We can't wait to discover what science will uncover next, and we want you right there with us. Until next time.